Okay, welcome to today's lecture. This lecture is the second lecture on human motor system, which is a part of the course autonomous robotics. Um, so last time we have go through the muscle structure, so the muscle autonomy and motor neurons and the circuitry in the spinal cord. And this is relatively lower level. And today we are aiming to go into the higher level to the brain. But before that, I would like to give you an intuition that how the outcome of this movement look like, which is the kinematics patterns. So we will first going to look at different kinds of movement and how this movement looks like. Because we are moving in the space, so we want to record the movement and see if there's regularity in the movement patterns. And we have a variety of movement from the normal movement in our daily life, like if I want to grab a cup of coffee or I walk, and even when I speak, I'm producing force in my jaw muscles. And when I look at you, I'm driving my eye muscles to navigate my gaze. And of course, there are more complex movements like in gymnastic and even in competitive sports, like if you can shoot a basketball accurately, you earn millions of dollars. So that is also very uh, impressive and competitive movements. But even with the most complicated movement, if we look at a smaller subset of this movement, it always composed of smaller primitives. Like we cut the movement, like for single movement, like reaching, and we can look at simplest case. So today I'm not going to to show you how the complex movement look like, but we start from the simplest case. So for example, like reaching point to point. So what about we just look moving from A to B? So this is a simple case that we can study. Like for example, if you grasp a pen and you move from one point to the target line, which is here, and you can observe that when you move faster, you become less accurate at the target, and if you move slower, you are more accurate on target. This is kind of intuitive, so it's easy to understand. And this is called a phenomenon is called speed accuracy trade-off. And this is actually an experiment that we can perform with the participant, and we can plot the data and to show some quantitative curves. Here they start from three different starting locations. So, and then at each start location, we can have different move times, like 140, 170, or 200 milliseconds. So the participants are instructed to finish the movement within this time duration. If they are too fast or too slow, the movement will not be considered. So in total, there will be 9 conditions, right? So 3 initial position and 3 movement times. And then, so in this case, so we have 9 conditions which represent by each of these 9 markers here. So we have a set of different movement time. So here, maybe the first 3 set corresponding to the, because it moves slower, so maybe corresponding to the closer movement, closer starting target, and the later 3 corresponding to the set of the further target. And we see again, this is a speed accuracy trade-off, right? So if you move the average speed is larger, then you will have this y-axis is the endpoint error. So you have larger endpoint error. So this is the speed accuracy, and this is relatively linear, right, across all the conditions. So it's a kind of a good fit to the experimental data. So this is just an illustration of this data for your more intuitive. And now let's move to a more complex case. So let's modify the task a little bit. So we can also change the width of the target. So that means this W here that you can say if I have a narrow width, then it's difficult to reach, right? If the wider is the easier task. And you can also change the A, which is distance between the start and end. So with this modification, so here let's say these variables are going to change. So we have the target width W and distance A. And in all conditions, they instruct you to move as fast as possible. So there's no you have to finish at a certain time. You just move as fast as possible. So this index with this log function of 2A divided by W represents how difficult is the task. So if the A is larger, so it's longer distance, but a more narrower target, so they will be more difficult, right? And vice versa. And again, if we recording the movement and plot these variables, we get, of course, after this logarithm, we get this more or less linear relationship between the difficulty of the task and the movement duration. So here we can fit in this linear equation with some offset and the slope. And this also like kind of a quantitative description of this between the speed accuracy trade-off. And this is called Fitts law. It's a very famous phenomenon in movement science. And let's go a little bit further to another type of movement. So before we always talk about point-to-point movement. So what about this circle movement? If you're drawing something like here, you have a like figure of 8, this kind of movement. You have to move around this curve. And so what we look at here is the curvature. Curvature by definition is 1 divided by the radius. So that means if you have, for example, this circle is large, so you have larger radius, then it will be the curvature here will be low. And what we look at the two variables is the velocity, which is the tangential velocity, which is the velocity along this direction on the curve, and the curvature c, which is 1 divided by radius. And here, for example, in the A example, so you draw this figure of 8, and what you see, the lower curve is the velocity and higher is the radius. So if you see if the radius is larger, for example, here you're moving here, the velocity is also large. And when the radius is smaller, the velocity is also slow. So here also, if you have this smaller circle inside, you also have this the same like larger radius or smaller curvature corresponding to larger velocity. So here again, you can do this curve fitting, right? So and in this case, will be fitted the curvature versus the velocity. And this is not quite linear, but you can do that as an exponential function. So that's why it's called power law. And this is kind of this continuous circular movement. Let's consider a more practical case. If you draw letter A, for example, this is also you have to go certain curvature. So here you have a few steps from 1, 2, 3, 4, 5, 6. So at the 2 position, it's relatively larger radius. So you have a smaller curvature. So with smaller curvature, we actually have larger velocity here. And also for the 0.4, right? So, but if you look at 5 and 6, so 5 and 6 is the reverse of the movement. So what is interesting here that by reversing, that means you have like a very smaller radius. So that's corresponding to an infinite curvature. So in this case, the velocity becomes slower in this case when the movement reversed. And this is more applied to a practical condition that you draw this letter A. And again, you have this, you can also map this to the power law in this case. Okay. And so this kind of regularity in this case. And here, let's look at more detail in the hand and joint. So this is also very classical experimental paradigm in research, which is 2D planar reaching. And the participant is grasping a handle and they can move this manipulandum like on a planar surface. So here they have 8 movement targets from T1 to T8 or T6, sorry, there's 6 movement directions. And so in all these conditions, you see the movement is relatively straight. So the subjects always tend to make straight movement. And if we plot the hand speed, so straight, I mean the hand is straight and the any factor. So if we plot the speed, it's kind of this bell-shaped, right? No matter which target you're switching between, the hand speed always bell-shaped. So that means you accelerate first and then you decelerate. You stop there. And this is also understandable. And if you look at joint angles, so we have elbow and shoulder joint. So the joint angle, the elbow and shoulder can have different profiles in time. while the hand speed is always similar, this bell-shaped. And also so this angle position and angle velocity, so they all depend on movement direction, but the shape of hand speed is invariant across different targets. And let's look again this hand path and hand velocity. So if we have different distance, so here in this case, you can have different velocity, right? So for larger distance, you will have higher peak velocity. But across all conditions, as long as point-to-point movement, there's always you'll observe this bell-shaped hand velocity. And of course, if you do the derivative, you get this biphasic acceleration. So this is a very like invariant, like observed regular pattern in human point-to-point movement.
Is the movement like forced, or can be like actively will another kind of movement?
This
like
is a
conscious,
this is conscious. This is a voluntary movement. You want to move from A to B.
But we can't force another shape. Like if you think about it.
Yes, you can force. So you can voluntarily change. But yeah, you're right. So it's not quite natural. So naturally, if you want to grab something, it will be like this.
Okay.
But of course, if you say if there's obstacle in front, so I have to this way, right? So you will not have this curve. So it is just a point to point. So this straight. Yeah. Yeah, of course, if you have more complex condition, you cannot get this. Yeah. Right, you're right. So yeah, this is the simplest case, I would say. Let's start from the simplest case. Then it comes to the modeling part of this curve. So as we know that we have this bell-shaped velocity profile. So if we can design some computer algorithms to predict this or to generate this, so here is a so-called minimum jerk model. So jerk is the 3 times derivative of the position, right? So one derivative is the velocity and twice is the acceleration. The derivative of the acceleration is called jerk. It's 3 dots here. And here in this case, the design is that if you can minimum the jerk cost, which is you integrate over the time. If you do this minimization, you get the solution for the result trajectory will be like this. So there's some complicated mathematical deviation, but I just show you the results. It will be like this. So here the condition is that you have you define the initial and final position, and you define the movement duration, and that's it. And using this algorithm, it automatically gives you this curve, this trajectory curve, which will be like this. So by minimizing the jerk function. Of course, this is is a kind of curve fitting. It doesn't tell you the mechanism. It just say that, okay, if we have the system, if they want to minimize jerk, then it will give you this curve, this smooth curve. Yeah. So then let's come to a more complicated condition. Again, this is a planar arm reaching. So they grab a handle. They move in the planar surface with 2 joints. So here we can do some tricks in this movement. So it's called null field. It means that there's nothing. It's just a natural movement. You see, if they go from the center, they can move to 8 targets. They always go straight. So what is interesting here? So if we can apply a perturbation, so the perturbation is applied on the handle. So there are some motors on this manipulator. They can at certain time they induce some force perturbation, and you can program that is as a force field. So here it is a velocity dependent force field. So let me explain what this means. So the force depending on the force amplitude and direction depending the hand velocity, which is the x and y direction. For example, if you have you are here, for example, you are moving x. For example, you are here, so you are moving the 0 x is 0, so y is some kind of maximum. So you're moving at one direction. And then you will have a perturbation on the right. So if you go straight, the force will drive you to the right. And similar if you do like here. So if you are here, so you are moving to the left, to the right, then the perturbation will drive you to towards you. So this kind of, it's very weird, this kind of prediction. But the point here is that they want to just induce some strange force field, like to challenge the participant, not to make natural movement. So what you see that at the first time when they induce this force perturbation, the participant cannot make straight movement anymore because as long as they move, the force field deviates their hand away from target. But later the participant realize, okay, well, I'm off the target, so I have to correct. So they somehow correct back to the target. So this is not very natural movement patterns. But subjects can learn, right? They can repeat. So the force field remains. So the participant just practice maybe 100 times against this force field. Then they learn to produce a straight movement again. So we call it, they adapt to this force field. So the adaptation is very similar to the case without this force field, so which is the straight line movement. And if we suddenly remove this force field, so it goes back to the null field condition, then there's after effect. That means the subject goes to the opposite direction. Even there's no force field, but it's just like this. So they go to the opposite direction. So it looks like the people, they have something programmed in mind and they still reproduce the same program with a force field. And then afterwards, of course, they can readapt, right? They can go back. But the immediate effect is that they produce this kind of movement. So this is adaptation. And the important thing is that even with the force field, after adaptation, the participants always trying to make straight movements. So it's kind of a natural iteration of when we produce kinds of movement. So here, let's sum up quickly about what we have learned. So we know this Fitts law, the speed accuracy trade-off, and this when we do this curvature drawing, we have this power law, and the bell-shaped velocity is always presented in the point-to-point straight movement, and we can use minimum jerk model to predict this trajectory. And with force adaptation, we see that after adaptation, participants always tend to reach the in the straight line. So this is just give you a roughly idea how we are not produce arbitrary movement, but there's some kind of regularity when we do this straight movements. And now let's come to a more difficult part, which is the brain, which is probably the most complex thing here. And okay, so this first is some terminology. So the central nervous system means the brain and spinal cord and then the rest is just a peripheral, including muscles and joints and so on. So now we are really talking about brain, and you see there are many structures that can be involved in this circuitry, and we will talk about later. And let's compare the roughly mass and body weight in different species. And we see humans kind of outstanding. So let me explain here. So if we start from lower level animals, you see their brain structure is very simple and the surface is very smooth, right? So in this fish and so on. And then if we go to the higher level species like the cat and the monkey and humans, you see this foldering, this called sulcus. So this they have this foldering inside this brain, and if you put all this foldering on the surface, it will be very large. And this is the advantage that they can save somehow save some space for the limited volume in human brain. And here we plot the mass, the brain mass versus the body mass in different species. And relatively, if you draw a linear curve and everything above this linear curve, that means the brain really have a large portion in the whole body, and the lower is less, the brain less. And apparently the octopus has possibly the largest slope here, and the chimpanzee and the human. And here dolphin is also kind of an outlier because it has very large mass, but still they have relatively large portion of the brain. So this is some data from. So let's come to more like neural stuff here. So that's what I want to talk about today, about very important brain areas that responsible for movement production. So I will mainly talk about three areas which may be most important for movement control, which is motor cortex, cerebellum, and basal ganglia. So here, before we go to these three parts, let's have an overview of this brain picture. So this is like a frontal plane. So if you cut in this way, the brain off, then you see in this intersection here. So on the surface, this folding is called cortex. Of course, there's not just motor cortex. Motor cortex is only part of the whole cortex. There's other part of cortex. So now we are only talk about motor cortex. There's also a premotor cortex. Sometimes people also call it together as motor cortex. So it's maybe just different terminology. And there's also a somatosensory cortex, which is receives the sensory information. And motor cortex gives more output to the motor system.
Does
Yep.
also sensory mean for touch? So it's
Yes,
touch?
yes, yes. So it's from the periphery and the sensory feedback. Yes, and also this like muscle spindle. This so it tells you your body position and so on. And inside the brain, you see there's a so cortex is only on the surface, right? And in between is the white matter, which is all these axons connections. If you still remember from last time. And here in the middle, there's a thalamus. This is also important because when the sensory signals come up from the spinal cord, all the sensory information must first stop at the thalamus, and then it goes out to other areas brain. The thalamus is kind of a relay of all sensory system, but we'll not talk about this today. And then there's a brainstem. So this midbrain and right nucleus. This is the one of the oldest brain structure. Like in lower species, they all have this brainstem. But cortex is really like relatively new to monkeys and humans. And on the side, there's this part is called basal ganglia. Though you see there are different components of the basal ganglia. We'll come to this later. And here, just below this, the whole big brain, and there's a smaller brain. It's called cerebellum. Why it's called a smaller brain? And because if you look at, they also have this cortex, right? This folder in here, and it's just like this, the bigger brain. So this is cerebellum is very particular, unique component of the brain, and it really receives all kinds of information, both sensory and motor. And we'll see later how what is structure of cerebellum and how it process this information. Of course, we don't know exactly yet, but I try to cover some findings till now. So here you can have a more fine division of the brain surface here. So this is a premotor cortex, and M1 refers to the motor cortex, or called primary motor cortex. The central sulcus separates the M1 and S1. S1 will be this somatosensory cortex. Okay, let's go a little bit deeper. So let's start from motor cortex. So this is the descending pathways from the motor cortex to the spinal cord. And we see it goes, there's one pathway going directly downwards from the motor cortex connect to the spinal cord. And this is called cortical spinal tract. So these are the axons, right? The cell bodies are located in cortex, and it synapses with the spinal cord, interneurons or motor neurons here. And same time, the motor cortex also sends descending tracks to other parts of the brain, including the red nucleus, which is part of the brainstem, and reticular nuclei also in the brainstem, and superior colliculus and vestibular system. So there are also other descending pathways, but we will not talk about this. And here, maybe we are familiar from last lecture that you have this alpha motor neurons, so the brain descending signal synapse on the motor neurons, and simultaneously they can also synapse through an inhibitory interneuron. And so there will be not necessarily all the direct connection. There could also some interneuron in between. And if we look at this is also like a horizontal cut of the spinal cord. You see the different descending pathways terminate on the different location of the spinal cord white matter, right? So you can corresponding all these tracks to these pathways. So this is the anatomy of the cortical descending pathways. So here, let's focus on, we will mainly talk about primary cortex because it's most studied and we believe it's most important for voluntary movement. And there are also other areas like the PMA, the premotor cortex, and SMA, supplementary motor cortex. So what will be the M1 look like? So if we cut in this way here, and then we get this area. So this is called a homunculus. Homunculus means the little man. So it's like, so you can actually map the different spot on the M1 surface to different body muscles. Like, for example, you have this leg. So in the top, you have this leg muscles, and then you have arm, trunk in between, and then arm. And what is very impressive that we see a very large area is dedicated to the fingers. It's from here, thumb to the little finger. It's a very large area compared to the whole. Because it's really small, but the control is very large. They occupy a very large area. And the same for the facial muscles here. You see, this is also large for the face muscles. And that's also intuitive, right? We are as human, we are particularly skillful at controlling these very fine motor movements. And you may wondering how this we get this. This is actually from the surgical experiment. It has been done a very long time ago. I think it's maybe 1970s. There's a famous Canadian surgeon called Wilt Penfield, and he actually mapped this homunculus when they do some operation. For example, they want to cut a tumor in the brain. They open the skull and they actually see the surface of the motor cortex, and they have to make sure that they don't cut the motor cortex. So they first check where it's important. So they put, for example, they put some electrodes here, and they stimulate this part, and they see a twitch on the wrist muscle. Okay, they know that okay, this part is responsible for this part, and they can actually map all this by stimulate individual spot on the surface, so they get this mapping. And interestingly, for the S1, we didn't talk about in the sensory map. They also have a very similar sensory homunculus, which have a similar distribution of this. So that means from sensory information, you also have a similar distribution. And maybe one point I want to add here is these eye muscles. It's not actually the control of the eyeball is not included here. It's another location called the frontal eye field, but it's not here. So just remember the when I control the eyeball is another location. But for the rest of the muscle, it's all shown here in the arm.
Well, it says eyeball there, so is it like conscious eyeball movement? Or...
Yeah, this eyelid and eyeball. I think maybe this is the sensory eyelid and eyeball. No, I think eyelid. No, I think yeah, maybe there's not right, but I'm sure that there's a frontal field. There's a particular other area that control the muscles of the eye. But this is maybe the eyelid is opening and closed. Yeah. Okay, so this is more like a scientific study about the neuron properties in the monkey M1 area. So this is again, this is 2D arm movement. The monkey need to navigate this joint stick to different directions on this planar surface. And here they record one neuron. For example, they just look at one neuron activity of one neuron in the M1. And then we can see that the neuron this neuron fires the most when it is 135 degrees. But if the monkey moves to other directions, the neuron have a very lower firing rate. So the same here, you see only at roughly this direction, they fire most, but while in the others, they fire less. So that means in one single individual neuron, you really cannot tell that move direction because it also fires in the neighboring direction. But if you do a population analysis, so here for each direction, so each pulse here is represented one neuron. For example, here you have 200 neurons, and since you have the tuning curve of each neuron, so at this particular direction, you can calculate the tuning direction, the firing rate of individual neuron. And once you sum all these 200 neurons, you get this red vector, which point roughly to the actual moving direction. So with this reconstruction technique, you can see a different direction. So only the population of the neuron can really tell you where is the direction. So that means the individual neuron really cannot tell you the movement direction. Only if you look at the population level, it makes sense. And this is actually a very famous study in M1 neuroscience. And again, when we talk about population neural activity in M1, we can also try to explain this drawing, like you draw a curve. So here we introduce a new concept called neural trajectory. So neural trajectory is a neural population activation at each time. So it's not just like average course. So in particular short time, you can calculate these firing patterns. And if we compare, so because this is 2D, right? So everything is mapped into 2D. And if you map neural trajectory with finger trajectory, you see it's actually very similar. Similar no matter where you move, right? And that means this movement plan is somehow represented by this population of neurons in the cortex. So this higher level planning gave rise to the lower level movement. And here, let's come back to this power law. As you remember, we can have this. So here, this is 2/3 power law. You can have different number here. But here, if you take 2/3 power of the curvature, you have relatively straight line in relationship to the angular velocity here when you do some drawing. And again, if we do this neural trajectory, you can have this similar mapping, and you can also do this technique to get this kind of this linear relationship that represents the movement plan. So, okay, so let's come to the next, the premotor area neurons, and this is have a different feature of the M1 neurons that M1 neurons directly related to the movement production. But here, if you see a monkey, so mostly we do this kind of experiment in monkeys. So if we record a PMA neurons, so before the movement, there's nothing here, almost nothing. And then when there's instruction to say, okay, move, then before the movement, this neuron already fires. But as long as the monkey starts to move, the neuron stops firing. So it seems that the PMA neuron is only like an indicator of the initiation of the movement, but it's not responsible for really producing the movement. And there's another interesting type in the PMA called mirror neuron. So what does the mirror neuron do? When the monkey picks up an object, a peanut, then the neuron fires. Okay, this is related to the actual movement. But if the monkey doesn't move and he observed another monkey do the same movement, pick up the peanut, the same neuron fires, even without any movement of this monkey. And also the same is also fires when a human do the same pickup of the peanut, it also fires. But if the human experimenter pick up the peanut with a tool, so that's kind of a different movement, then the neuron doesn't fire. So in this experiment, it seems that this mirror neuron fires both when the monkey do the experiment and they observe the other monkey does the experiment. Okay, so that is about the motor cortex. Let's go into this cerebellum. It's quite exciting brain area because it's really, you see the cerebellum is very unique. It's located like below the main part of brain, and it receives all these motor information. Later we will see it receives all this motor information and sensory information. So everything put together feed into the cerebellum. And actually, without cerebellum, you can still able to move, but you will observe some abnormal way will look like later. And cerebellum, people say that it's like a conductor. So if it organizes everything, but it doesn't produce a movement that is modulated and organized. Like if you have in an orchestra, like he can command, okay, this violin plays and then the drummer plays. So they kind of say, okay, which time you active and you active. But without cerebellum, they can still, the component is still playing, but they cannot synchronize to do a coordinated way. So that is roughly what the cerebellum is doing. And anatomically, you see the cortex, the motor cortex send commands first through the pons, which is the area of the brainstem, and then these motor commands going to the cerebellum, and these go to the ventrolateral. This is a part of the thalamus, and then it goes back to the motor cortex. Also, you can see here the cerebellum also the output of the cerebellum. First, the input is here, right, from the cerebral cortex going to cerebellum. And the output, first they go to the red nucleus and go to the spinal cord, and also through the thalamus go to the motor cortex and go to the spinal cord. So this is the anatomical loop in the cerebellum. So let's look a little deeper in the neural circuitries in the cerebellum. Maybe let's first forget about all this terminology of different fibers. But roughly speaking, so the cerebellum, first you see these arrows going up. This is the input to the cerebellum. You see it terminates. First, this is the deep cerebellum nuclei. So this is kind of a center processing location of the cerebellum. And it receives the input from both sensory and motor signals. And same time, these signals travel all the way up, connect to like they call it a parallel fiber. Like they go this surface. And this parallel fiber synapse with the very large, this is called Purkinje cell. So they synapse to the dendrite of the Purkinje cell. And Purkinje cell is the only output from the cerebellar cortex that directly goes to the deep cerebellar nuclei. And this deep cerebellar nuclei send the final command to the motor cortex through thalamus. So this is why this is interesting, because this structure is repeated all the way through the parapolar cerebellar cortex. So this is just one unit. If you imagine that you have this one cell and then just repeat it, all the same structure. And of course, this different area may correspond to different body segments, but the structure is always there. And this is also fascinating the scientists a little bit, why it's so highly structured cerebellum. But still, we don't know how exactly it functions. But we know, currently we know roughly is that it takes all the motor information and sensory information and send out a modulation command to the motor cortex. And actually, there's some hypothesis about why cerebellum is important. So people think cerebellum is involved by feedback error learning, because you have the motor information which means that what you intend to do, right? And you get the sensory information, that is what's the result. And if you compare these two, the difference will be that whether if the difference is zero, then there's nothing, it's okay, everything is fine. There's an error that tells you, okay, you have to correct this. And only if you have an error, cerebellum is involved. If everything is perfect match, the cerebellum is not doing anything. So the engineers propose some control scheme here, like this feedback control stuff. And they also map different so-called internal models to different cerebellum areas, like the cortex and mostly fibers and so on. But we will not go to the details. These are also some more computational studies, like they can do some models and to control a plant. And I want to tell you what happens if the cerebellum has a deficit. For example, in patients, one thing you observe is that the movement delay. So if you by the way, the cerebellum is always ipsilateral. That means if you have lesion in the left cerebellum, your left part is is affected, but it's opposite for the cortex. For the cortex, if your left part is defective, your right part is disabled. So it's called contralateral. But for cerebellum, it's ipsilateral. So if you have left lesion in cerebellum, then you observe that the left hand is when you simultaneously produce movement, the left hand has a certain delay. Also, movement errors. If you pointing somewhere, you will see the patient will not arrive smoothly at target, and they even not just this curved movement, they always either overshoot or undershoot the target. So they just cannot arrive at the perfect arrive at the target. And if you do a patterned movement error, they also cannot do this synchronized periodic movement. Here, this is called interaction torque in this case, because if you want to lift your elbow, but without moving your shoulder, this movement will cause interaction torque on your shoulder. So you have to somehow compensate the interaction torque from the elbow. And here in the control, which is the normal participant, you see this, the net, this blue is the net torque is kind of zero, and even you have interaction torque here is compensated by the control participant. But in the cerebellar patient, they cannot compensate for this interaction torque. You see this large net torque here in this blue curve. That's result a movement of the shoulder. So they cannot really compensate for this interaction torque. This is also a typical phenomenon in cerebellar patients. Okay, let's quickly come to the next brain area is basal ganglia. Here we can see roughly the connection of the basal ganglia. It also receives input from motor cortex, and it sends out the output back to the motor cortex. So it's kind of a loop. And let's go into a deeper look at how this loop is formulated. And we see in this painting, they have several components of the basal ganglia. And you don't need to remember all these names, but roughly there's a striatum which consists of caudate and putamen. And this corresponding to these two parts, but they're actually connected, but it's not shown here. And another part called the pallidum here. We'll come back into next slide. And this subthalamic nucleus is considered to be part of the basal ganglia, but actually it's also part of the thalamus. That's why it's called kind of thalamic here. And important thing is here, I want to mention, it's a substantia nigra. It's a location where it's produced a neurotransmitter called dopamine. And this is very important for movement initiation. And we will see later what happens if this part has a problem. And let's see the movement, this neural circuitry in the basal ganglia. So this actually quite complicated. So you see different loops that go through the motor cortex and through the basal ganglia, and then it also goes back to the motor cortex through thalamus. So here is a direct and indirect loop. So I think I will not go to because this is very detailed. So we just need to know that there are two pathways, direct pathway and indirect pathway. So indirect pathway, they go through more relays. And so here I want to say again, this substantia nigra is the source of producing neurotransmitter dopamine. And we'll see how this works. So here again is the same loop before, but it just shows you the different neurotransmitters. They have different type like glutamate and GABA and dopamine. So dopamine can be both excitatory and inhibitory. So depending on the type of the dopamine receptors, for example, here you have D1 and D2, and depending on which type of the receptor, it can produce different effect of the subsequent loop. And here, I don't want to go to the details of this loop. It just let you know that we have different type of receptors, and this dopamine is really what the mainly the basal ganglia produce to drive this movement. So here, again, this direct, we have talked about direct and indirect loop. So the direct pathway goes through the caudate putamen and goes to pallidums and go back to the cortex through the thalamus. While for the indirect pathway, they have to go to more complicated loop. And the point here that is that we need to notice there's a plus and minus sign here, which means the excitation or inhibition. And depending on how many stations they go, so you can just do a calculation to, like, for example, a plus minus minus plus will be plus, right? So this loop will be excitatory. And if you take another loop, for example, here, it's plus minus 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 plus, they will be inhibitory. And in phasogania loops, there are both excitatory and inhibitory loops, and they can call it indirect or direct. So the net effect is always a combination of the both loops. And we will see later what happens if this balance is destroyed. So this will be result in different diseases. So the first disease we look at is the Parkinson disease. And what happens here is there's a here, so this part from the caudate and putamen to the pallidum, so this part is affected and it has an uh, increased activation here. Then this whole loop, so if you calculate this plus minus, the net effect will be the more inhibition to the cortex. So which means in Parkinson's disease, the net effect of this lesion will result in inhibition of the motor cortex. That means it inhibits your movement. And that's what you observe in the Parkinson's disease. They have problem in initiating the movement, but they have also some side effect. It's typically like they have the resting tremor. They cannot move, but they freeze and they have some tremor. But as long as they somehow able to move, the tremor disappears. And the cause has been identified as the loss of dopamine neurons will take effect here. So the degeneration, you see that I just mentioned that the substantia nigra produce this dopamine. If this is degenerated, they will have this result. And we don't know why such neurons die. So we still don't know the mechanism of this disease. And then the opposite way is called Huntington disease. So what happens here is, so in contrast to the decrease, this loop is an increase, and here this decrease. So the net effect in Huntington's disease is that they have less inhibition. That means they have more excitation to the cortex. And what it will result is more movement. So if you don't want to move, it just excites cortex and you move. And that's why you always see this dancing effect. They're playing involuntarily their limbs. And yeah, but they still coordinated. Probably the cerebellum is taking charge of this. And the cause somehow identified by the gene mutation for this patient. Okay, so I think we are close to the end, but I want to show you a video. Do this. How to, okay, of this Parkinson disease. So the problem is here from neurally is a degeneration of dopamine allergic neurons in substantia nigra, and you see it has a significant tremor in the limbs, and he's very hard to initiate a gait. So only with help of the clinician here. And it's really stumbled here. And finally, he managed to do that. I'll go a little bit through. So here, what about if you let him run a bicycle? So as long as I push it, this just automatically drive this bicycle. So it seems once, as long as the movement is initiated, it has no problem in doing this. And he's also able to look around and make a turn. So in this case, it seems everything is normal in this person. And then he can back to, and as long as he stepped out, it's still freezing here. You see, he cannot move. Yeah, that is what you see what happens with these Parkinson patients. And we know that we have identified the problems here, but still there's a lot of things to study. Okay, so let's summarize. So we have learned that motor cortex is involved in the voluntary movement, and the cerebellum is responsible for coordinating the movement. And basal ganglia also, they have a lot of connections with movement regions, and particularly if something goes wrong, what will happen in these patients. Okay, this is think this is the end of this lecture. So let's say we have learned this muscle. We start from this muscle and reflex loops, and we see the regularity in movement patterns, and we also see today these different brain regions. So I hope you get an overview of how this motor system is structured. Of course, we cannot really go to all these details. There's a lot of like textbooks that if you are interested, you can dive in this. A lot of findings, but still a lot of unknowns, a lot of research are going on in this area. Yeah, so that is, I think that is for today. Stop.